This method uses fluorescence to determine if a protein transfers two biologically important lipids between synthetic membrane and thus contribute to the distribution of these lipids inside eukaryotic cells.
This technique makes it possible to readily measure the extraction and transport of a natural lipid by a protein. It requires standard equipment and fluorescence sensors and liposomes that are easy to make. This method can be used and modified to get insight into cellular biology by unveiling the proteins behind the distribution of some essential lipids between organelle membranes. Visual demonstration is critical to explain how to perform real-time measurements of lipid transfer by fluorescence.
So demonstrating the procedure will also be Maud Magdeleine, an engineer from my laboratory.
Begin by preparing fresh, filtered, and degassed Hepi's potassium acetate buffer supplemented with 1 mmol magnesium chloride to form HKM buffer. Then, prepare liposomes only made of PC or additionally doped with 2 molar % PS or PI4P. Now, place the flask on a rotary evaporator to dry the lipid under vacuum. In one well, mix liposomes containing 2 molar % PS with lipid sensor NBD-C2 lact to a final concentration of 250 nanomolar in a volume of 100 microliters. Fill a second well with the same amount of liposome and NBD-C2 lact mixed with 3 micromolar lipid transfer protein, or LTP. Fill a third well with 250 nanomolar NBD-C2 lact mixed with pure 80 micromolar PC liposomes and a fourth well with pure 80 micromolar PC liposomes.